Alright, in this unit we're going to learn about DNA and then how DNA is used to make proteins. Um, first we have to learn about the structure of DNA. So DNA is short for deoxyribose nucleic acid. Alright, it's called deoxyribose because it has a sugar called deoxyribose in it, and it's called a nucleic acid because that's what it is. So remember back to our organic molecules. Nucleic acid is one of them. So the type of organic molecule that DNA is a nucleic acid. Nucleic acids are the DNA is found in the nucleus of eukaryote cells. Remember, in prokaryote cells, they don't have a nucleus, so it would be just found in the cytoplasm. We're just going to focus on eukaryote cells for this section. And then in the nucleus, DNA is found on chromosomes, and we're going to talk more about that in the next section. So, DNA so that's just some background. So we've talked about pretty much all these things. We talked about chromosomes and mitosis when they divide. But now we're going to get into the structure of DNA. So DNA, you guys probably all know this stuff. It looks like a twisted ladder, goes around in like little circles, and that's called a double helix. So it looks like a twisted ladder, and its shape is called a double helix. And the reason why it's called a double helix is because there are two strands of DNA twisted together. And so here we have one strand going like this, and we have the other strand going like this. So what I want you to do is add a picture here. Now, another review is when we were talking about the organic molecules, we talked about nucleotides. Nucleotides are the building block of DNA, and nucleotides are made up of a sugar, that's the house. You got the base, and the base can be coming off the side instead of coming up the top. And then you got a phosphate in the circle. Alright, so this is important. You need to recognize that a nucleotide is made up of those three things. So once we finish putting these down, you're going to have to put another picture of a nucleotide here and make sure you identify the three things. And remember the sugar is called deoxyribose. And then again, put a picture of a nucleotide here and label the three parts. So again, that should be review. So now we're going deeper into the structure of DNA. And here it is. So here's one strand of DNA, and here's the other strand. The only thing that we did is we straightened out the DNA instead of having it twist. So each strand of DNA is connected by the sugar and the phosphates. So they make the outer ring of DNA. So if you look here, the sugar, phosphate, sugar, phosphate, sugar, phosphate. So we call these the backbones because they're always on the outside, and it's always the same thing. As you see, it just repeats. So this isn't the most important part of DNA. The most important part of DNA is the bases. So in the middle, there's these bases here. So you have A pairs with T, G pairs with C, which we'll talk about in a second. And the order of these bases determines our genes, which again we'll talk about later on. So the bases go in the middle, the nitrogen bases. And again, here's our whole nucleotide. Here's a nucleotide. Here's a nucleotide. Over here, here's a nucleotide. They're just upside down. But here's a nucleotide, so it's the same thing repeated over and over. The only thing that's different is which base is on that nucleotide. So if we go back to the notes, each strand of nucleotides is connected by a sugar and phosphate, and the backbone of DNA is made up of a sugar and phosphate. The two strands are connected by bases, the nitrogen bases in the middle, and those bases make up the rungs of the ladder. So if you think about the middle, the stuff that you actually climb up would be the rungs here, and then these are the sides of the ladder. So now the bases are important, so let's talk about them. So there's 4 bases. And again, draw that picture. Those are right on that slide, because that's going to be very important for what you're going to do next. The bases, whoops, this should be there. Or the bases are the most important, part of DNA. And there are 4 bases. You have adenine, which is A. We're mainly just going to call them by the letters. Thymine, which is T. Guanine, which is G. And cytosine, which is C. And then with this, if we go back here, A always pairs with T. So wherever there's an A, there's always going to be a T. Wherever there's a C, always pairs with a G. So you can remember that by A and T spell at, and G and C don't spell anything. Or you can also remember A and T have straight lines, G and C are curved. So A always pairs with T, and G always pairs with C. Alright, so that's going to be very important. You're going to have to know those base pairing rules. So if I give you a segment that says it is, so if I give you a segment that it is A, T, T, G, C, G, you're going to need to tell me the complementary strand, which is the opposite strand. So the opposite strand of that would be T, A, C, G, C. Alright, so that is how base pairing works. Alright, now the sequence of the bases make up a gene. So a gene is the order of those bases. And we can have a different order. So an order for unattached earlobes might be one segment of, might be ACT, GAC, that's a little bit small. But then the other one might be ATC, GACT. So just the sequence of bases just determines whether or not your earlobes are attached or not, and that goes for a lot of genes. And we're going to talk more about that in the next unit. Right now you just need to know that the sequence of bases make up a gene. Alright, and then that sequence ultimately then will determine our trait. So the sequence of bases make up a gene, and then the gene determines our proteins. A lot of people like to say it's a trait, but it actually determines our proteins, which later on determines our trait, and then the proteins determine our trait. So down here I just want you to put this part, DNA, arrow, gene, arrow, protein, trait, because that's the order. DNA, the sequence of DNA determines a gene. The gene determines a protein, and this is the part that we're going to focus on in this unit, how a gene is transferred into a protein, and then the protein, the trait. So we're going to talk about more of the traits in the next unit. Alright, so again, if you have a different sequence of DNA, you might get one gene. So you could have 2 different sequences, one for unattached and one for attached, so those are your 2 different genes. They make 2 different proteins, and that's why either your ears are attached or unattached. So that's what you need to know. Again, you need to know, this is probably the most important picture for you to know, because it tells you the base pairing rules, and the sugar and phosphates on the outside shows you nucleotides, so this is probably the most important thing. And then you'll also definitely need to know this sequence.